Every second, your cells are making a choice: produce energy and thrive, or falter and age. Deep inside, mitochondria, a name you might remember from a textbook, are quietly shaping the course of your life, from your first breath to the subtle slowdowns of age. What if the tiny engines inside your cells were not just background machinery, but central to why you feel tired, why your muscles ache, or even why memory sometimes slips? You will discover how these powerhouses really work, why their efficiency changes as decades pass, and what happens when their energy output declines. Most intriguing of all, there are emerging strategies, some surprisingly simple, that might help you recharge these engines, possibly slowing aging itself. Once you see how much rides on mitochondrial health, you will never look at fatigue or vitality the same way again, and you will want to hear every secret these tiny organelles have to offer. Picture for a moment the bustling interior of a living cell. Within this microscopic metropolis, countless processes are in perpetual motion, each one vital to life itself. At the heart of this activity, orchestrating the cell's energy needs, are remarkable structures known as mitochondria. These organelles, often described as the powerhouses of the cell, are foundational to the existence of complex life, quietly enabling every heartbeat, every thought, and every breath. Mitochondria are found in nearly every eukaryotic cell, meaning they are present in the plants, animals, fungi, and protists that make up much of life on Earth. Their ubiquity is no accident. They produce about 90% of the energy that cells require, a staggering contribution that underscores their essential role. Without mitochondria, the energy demands of complex multicellular organisms could never be met. Structurally, mitochondria are small, oblong organelles, typically measuring between half a micrometer and 1 micrometer in diameter. Their most distinctive feature is a double membrane system. The outer membrane encases the organelle, while the inner membrane is intricately folded into structures called cristae. These cristae are not merely architectural flourishes. Their folds provide a vast surface area for critical energy-generating reactions, allowing mitochondria to maximize their output. Nestled within the inner membrane is the matrix, a gel-like space that houses the mitochondria's genetic system and the enzymes responsible for breaking down nutrients. The story of mitochondrial energy production begins with the nutrients we consume: carbohydrates, fats, and proteins. Once inside the cell, these molecules are broken down, with the final products, such as pyruvate from glucose, entering the mitochondria. The matrix becomes the stage for the citric acid cycle, also known as the Krebs cycle, a series of enzyme-driven reactions that further dismantle these molecules, extracting high-energy electrons in the process. This is only the prelude. The real magic happens along the inner membrane, where oxidative phosphorylation takes place. Oxidative phosphorylation is a marvel of biological engineering. High-energy electrons, harvested from earlier stages of metabolism, are passed along a chain of protein complexes embedded in the inner membrane, collectively known as the electron transport chain. As electrons move down this chain, they drive the movement of protons, positively charged particles, across the membrane, creating a proton gradient. This difference in proton concentration across the inner membrane forms a reservoir of potential energy. It is this gradient that powers ATP synthase, a molecular machine that generates ATP, or adenosine triphosphate. ATP is often called the molecular currency of energy. Its structure, a molecule composed of adenine, ribose, and 3-phosphate groups, may seem simple, but it holds within its bonds the energy that fuels virtually every cellular process. Breaking the high-energy bonds between phosphate groups releases energy that powers muscle contractions, nerve impulses, and the synthesis of essential biomolecules. Incredibly, the human body is estimated to produce around 65 kilograms of adenosine triphosphate each day, recycling each molecule thousands of times to maintain this furious pace. The adenosine triphosphate produced in mitochondria is not trapped within the organelle. Instead, it is pumped out into the surrounding cytoplasm via an adenosine triphosphate/adenosine diphosphate shuttle system, making it available wherever energy is needed. This seamless delivery ensures that processes requiring immediate energy, from the beating of a heart cell to the firing of a neuron, can proceed without delay. The efficiency of this system is remarkable. Mitochondria can generate about 15 times more adenosine triphosphate from the metabolism of sugars than glycolysis alone, the process that occurs outside mitochondria in the cytoplasm. Not all cells have the same energy needs, and mitochondria respond accordingly. Cells that demand a lot of energy, such as those in the heart, muscles, brain, liver, and kidneys, are packed with mitochondria, sometimes numbering in the thousands. In contrast, cells with lower energy requirements may contain only a few. Mitochondria are dynamic, reshaping themselves, fusing together, or dividing as needed to meet the cell's fluctuating demands. They can even localize to regions within the cell where energy consumption is especially high, ensuring that adenosine triphosphate is supplied precisely where it is most needed. One of the most intriguing aspects of mitochondria is their genetic independence. Unlike most organelles, mitochondria possess their own deoxyribonucleic acid, a small circular chromosome containing 37 genes. This deoxyribonucleic acid encodes essential components of the mitochondrial machinery, including 13 proteins central to oxidative phosphorylation, as well as transfer ribonucleic acids and ribosomal ribonucleic acids needed for protein synthesis within the organelle. However, the vast majority of mitochondrial proteins, numbering in the thousands, are encoded by the cell's nuclear deoxyribonucleic acid and imported into the mitochondria. This dual genetic system is a relic of their ancient origins. The prevailing view among scientists is that mitochondria descended from free-living bacteria that entered into a symbiotic relationship with the ancestors of modern eukaryotic cells. Over millions of years, this partnership became so intimate that the bacteria surrendered much of their autonomy, sharing their genetic material with their host. The remnants of this ancient merger are evident today. Mitochondria divide independently of the cell, possess their own genome, and even share similarities to bacteria in their structure and reproduction. This endosymbiotic origin was a turning point in the evolution of life, enabling the complex multicellular organisms we see today. Mitochondria are more than just energy factories. They are social, dynamic participants in the life of the cell, communicating with each other, fusing to share contents, and dividing to distribute themselves as needed. Their influence extends well beyond energy production. They help regulate cell death, a process known as apoptosis, and play a role in aging and cellular signaling. The breadth of their impact is still being unraveled, but it is clear that mitochondria are central to the well-being and survival of the cell. There is, however, a price to pay for such prodigious energy production. As mitochondria convert oxygen and nutrients into adenosine triphosphate, they generate byproducts known as free radicals, reactive molecules that can damage cellular components, including mitochondrial deoxyribonucleic acid. To counteract this, mitochondria are equipped with antioxidant enzymes such as superoxide dismutase, which help neutralize these potentially harmful species. Despite these defenses, mitochondrial DNA remains particularly vulnerable to damage, in part because it is located so close to the site of free radical production and lacks many of the protective mechanisms found in nuclear DNA. Over time, the cumulative effects of this damage can contribute to cellular dysfunction and aging. As mitochondrial efficiency declines, so too does the cell's ability to produce ATP, affecting tissues and organs that are especially dependent on a steady energy supply. This decline is a subject of intense scientific study, as researchers seek to understand how mitochondrial health influences the aging process and the development of diseases. Despite these challenges, the resilience and adaptability of mitochondria are nothing short of extraordinary. Their ability to respond to changing energy demands, to repair themselves, and to communicate with other cellular components speaks to their enduring importance. As science continues to probe the mysteries of these tiny powerhouses, one truth remains clear. Mitochondria are not only engines of cellular life but also guardians of its vitality, shaping the fate of every cell and, by extension, every organism. Their story is a testament to the intricate choreography of life at the most fundamental level and to the remarkable systems that drive it forward, one molecule of ATP at a time. From the earliest moments of life, our cells are powered by the silent, ceaseless work of mitochondria. These tiny, ancient organelles do more than fuel our existence. They are constantly adapting, dividing, and communicating to maintain harmony within each cell. Yet, the journey from youth to old age is marked by profound changes in how efficiently our mitochondria work and how well they protect themselves from harm. The story of mitochondrial function across the lifespan is not just one of gradual decline, but of dynamic change, intricate quality control, and continual negotiation with both our genes and our environment. At the heart of this story is mitochondrial biogenesis, the process by which new mitochondria are formed within our cells. This is not a simple act of duplication. Rather, it is a tightly regulated symphony of nuclear and mitochondrial genes working together. Cellular signals trigger the synthesis of proteins, import them into the mitochondria, and replicate the mitochondrial DNA itself. This careful choreography ensures that as cells grow, divide, or respond to increased energy demands, they maintain a healthy population of these essential energy converters. But mitochondria do not simply multiply unchecked. Their numbers, shapes, and locations are under constant surveillance. Mitochondria can split a process called fission or merge with one another, known as fusion. This dynamic behavior allows the mitochondrial network to adapt quickly to stress, redistribute energy where it is needed most, and dilute damage that might accumulate in a single organelle. When mitochondria become damaged beyond repair, they are selectively removed by a process called mitophagy, a specialized type of autophagy that recycles their components for reuse. These quality control mechanisms are crucial not only for maintaining energy production, but also for cellular survival, especially in organs that demand high continuous power like the heart and brain. As we age, however, the efficiency of these processes begins to wane. Mitochondrial DNA, distinct from the nuclear genome and inherited solely from our mothers, gradually accumulates mutations and suffers from oxidative damage. Over time, this erodes the integrity and function of the mitochondrial genome, leading to a measurable decline in both the volume and quality of mitochondrial DNA within cells. The mechanisms responsible for synthesizing new mitochondrial proteins also lose their vigor with advancing years. In human skeletal muscle, for example, the rate at which both contractile and mitochondrial proteins are produced diminishes, which in turn alters the muscle's metabolic capacity and contributes to the weakness and fatigue that often accompany aging. The consequences of these age-related changes ripple outward, affecting entire tissues and organ systems. In the brain, declining mitochondrial function is closely linked with reduced synaptic activity and increased production of reactive oxygen species, unstable molecules that can further damage cellular structures. Such dysfunction is particularly evident in conditions like Alzheimer's disease, where the decline in mitochondrial quality and activity is associated with cognitive impairment. Similarly, in the heart, the turnover and repair of mitochondria become less efficient, contributing to the gradual loss of cardiac function that is seen with aging. Underlying these visible effects is a complex network of molecular quality control. Mitochondrial health is preserved through a combination of biogenesis, fission, fusion, and mitophagy, all orchestrated to balance the production of new organelles with the removal of damaged ones. Cells also deploy additional quality control systems to monitor the health of mitochondrial proteins and organelles, sometimes marking defective components for degradation before they can inflict harm. These processes are so fundamental that they have become targets for therapies aimed at slowing the aging of the heart and other organs. Yet, the story of mitochondrial decline is not written solely in our genes or the relentless march of time. Environmental factors and lifestyle choices can exert a powerful influence on mitochondrial health throughout life. For instance, exposure to pollutants, tobacco smoke, or certain pesticides has been shown to disrupt mitochondrial function and impair the quality control systems that keep these organelles running smoothly. Even common substances like caffeine can have measurable effects on mitochondrial performance, although the precise consequences may depend on dose and individual biology. Stress, too, can reshape mitochondrial metabolism. In vascular cells, environmental stressors can alter the way mitochondria process energy, influencing the growth of new blood vessels and the overall metabolic health of tissues. When the balance tips too far, when stress is chronic or environmental toxins overwhelm the system, mitochondria may suffer damage to their membranes and DNA, triggering a cascade of cellular dysfunction that contributes to disease. The interplay between genetics and environment is particularly evident in the inheritance of mitochondrial DNA. Because all our mitochondrial genes are passed down from our mothers, variations in this small genome can influence traits such as height, lifespan, and susceptibility to diseases like type 2 diabetes and multiple sclerosis. Research suggests that the expression and maintenance of mitochondrial DNA can be shaped by lifestyle factors and environmental exposures, further linking the fate of our mitochondria to the world we inhabit. Across the lifespan, the highly plastic and dynamic nature of mitochondria allows them to adjust to changing metabolic needs and environmental challenges. They adapt by altering their number, shape, and position within cells, and by engaging in fission, fusion, and selective removal of damaged units. However, as we grow older, the cumulative effects of DNA mutations, oxidative damage, and declining quality control gradually erode this resilience. The result is a progressive loss of cellular function and energy production, a hallmark of aging that is felt in every tissue and organ system. Despite these challenges, there is evidence that certain lifestyle interventions can slow the decline in mitochondrial function. Nutrition, physical activity, and even caloric restriction appear to influence the health of mitochondria and may help extend the healthy years of life. These interventions likely work by supporting the very quality control systems, biogenesis, fusion, fission, and mitophagy that preserve mitochondrial integrity. Of course, many mysteries remain. While the molecular machinery behind mitochondrial quality control is increasingly well understood, the precise ways that environmental factors, genetics, and daily choices combine to shape mitochondrial health across a lifetime are still being unraveled. What is clear, however, is that the fate of our mitochondria is inseparable from the fate of our cells, and by extension, from our own vitality and longevity. In the end, the journey of mitochondria from youth to age is a story of dynamic adaptation and gradual compromise. It is a testament to the remarkable resilience of life at the smallest scales and a reminder that our energy, our health, and our future are intimately tied to the fortunes of these ancient organelles. Through ongoing research and thoughtful stewardship of our environment and lifestyles, we may yet find ways to support mitochondrial function and, perhaps, slow the inexorable passage of time within our cells. The slow, relentless ebb of cellular energy is not simply a matter of running low on fuel. Instead, it is a complex symphony of molecular events, many of which can be traced back to the mitochondria. At the heart of this process lies a paradox. The very organelles that power life through energy production are also vulnerable to the forces that ultimately constrain it. Over the decades, the delicate machinery inside mitochondria faces a cascade of challenges: oxidative stress, genetic mutations, and impaired internal dynamics, each subtly eroding their vitality and, in turn, the health of the entire organism. One of the most persistent threats to mitochondrial health is oxidative stress. Within the bustling interior of a cell, mitochondria are the main generators of reactive oxygen species, or ROS. These chemically reactive molecules are natural byproducts of breathing and energy generation, but in excess, they become agents of chaos, attacking proteins, lipids, and the very DNA that guides mitochondrial function. Evidence shows that most oxidative stress in cells is actually generated by mitochondria themselves, and as this burden grows, so does the risk of damage. What starts as a trickle of molecular injury can, over time, become a torrent, overwhelming the cell's defenses and spreading dysfunction throughout the mitochondrial network. This oxidative assault does not occur in isolation. Mitochondria contain their own DNA distinct from the DNA distinct from the DNA found in the cell's nucleus, and this mitochondrial genome is especially vulnerable to oxidative damage. Unlike nuclear DNA, which benefits from robust protective and repair mechanisms, mitochondrial DNA is exposed, almost defenseless, to the barrage of ROS. The consequences are profound. When mitochondrial DNA is damaged, errors in replication and gene expression can follow, disrupting the finely tuned production of proteins essential for energy metabolism. This, in turn, impairs the mitochondria's ability to generate ATP, the currency of cellular energy, setting up a vicious cycle. Impaired function leads to more ROS production, which causes further damage, and so on. Genetic mutations, whether inherited or acquired, further complicate this picture. Mitochondrial diseases, genetic conditions that directly affect the organelle's ability to produce energy, are estimated to impact about 1 in 5,000 individuals. The symptoms are strikingly varied, ranging from muscle weakness and neurological deficits to heart dysfunction, reflecting the central role that mitochondria play in tissues with high energy demands. But mutations in mitochondrial DNA are not only a matter of rare diseases, they also accumulate gradually in everyone as they age. Over time, these mutations can reach a threshold that tips the balance, manifesting as clear declines in energy and cellular function. Research indicates that a certain proportion of mutated mitochondrial DNA must be present before these changes become apparent, showing that cells are resilient up to a point. But beyond that, dysfunction rapidly accelerates. The implications of mitochondrial dysfunction extend far beyond fatigue or loss of vigor. A growing body of evidence points to these molecular breakdowns as pivotal events in the development and progression of a wide range of chronic diseases. In neurodegenerative disorders such as Alzheimer's disease, the consequences of mitochondrial decline are particularly devastating. Damaged mitochondria in neurons lead to deficits in energy metabolism, disrupt protein processing, and ultimately promote cell death. This cascade not only interrupts communication between nerve cells, but also amplifies other pathological changes characteristic of neurodegeneration. Oxidative stress and mitochondrial dysfunction reinforce each other, making it difficult for neurons to maintain the energy necessary for basic function, let alone repair or adapt to ongoing damage. Similarly, in metabolic diseases such as diabetes, the same patterns emerge. Mitochondria become less efficient at producing energy, while increasing levels of ROS damage cellular components and disrupt metabolic signaling. The result is a gradual decline in tissue function, impaired glucose metabolism, and, eventually, the emergence of systemic symptoms. Cardiovascular diseases, too, are closely linked to mitochondrial dysfunction, with the heart's enormous energy needs making it particularly susceptible to any shortfall in ATP production. Even cancer is not immune from the far-reaching influence of mitochondrial decline. Some forms of cancer are associated with mutations in mitochondrial DNA that disrupt the normal process of oxidative phosphorylation. This not only alters the energy landscape of the cancer cell, but also affects the regulation of cell death, potentially allowing damaged or abnormal cells to survive when they otherwise would not. The interplay between mitochondrial dysfunction, energy deficits, and the breakdown of cellular quality control is a recurring theme in the story of chronic disease. What then drives this decline in mitochondrial health? The evidence points to a convergence of factors. Aging stands out as a major contributor. As years accumulate, so do genetic mutations and oxidative damage, gradually eroding the efficiency of the mitochondrial network. Inflammation and poor metabolic health also take a toll, increasing oxidative stress and compromising the repair and maintenance of mitochondria. Environmental stressors, ranging from pollutants and toxins to infections, are now recognized as powerful disruptors of mitochondrial function. In fact, mitochondrial dysfunction is increasingly seen as a hallmark of environmental injury, with pollutants and toxins capable of sabotaging the organelle's intricate processes and accelerating the march toward disease. The consequences of cellular energy decline are not limited to the microscopic world of molecular machinery. They reverberate outward, shaping the trajectory of aging and the onset of frailty. As tissues lose their capacity to generate energy efficiently, the body as a whole becomes more susceptible to dysfunction, inflammation, and disease. Aging is now understood, in part, as the story of accumulated cell damage and declining function, with mitochondrial dysfunction playing a starring role. Oxidative damage to mitochondrial DNA, in particular, impairs the replication and expression of genes critical for cellular metabolism, creating a feedback loop that hastens the decline. Perhaps most strikingly, the scale of mitochondrial activity is almost unimaginable. In a single day, the human body produces its own weight in ATP, a testament to the ceaseless work of these organelles. In some cells, mitochondria can occupy up to a quarter of the entire cell volume, underscoring their centrality to life. When this engine falters, the effects are systemic, fatigue, cognitive impairment, muscle weakness, and the gradual unraveling of complex physiological systems. It is also becoming clear that mitochondrial dysfunction does not occur in isolation. Instead, it is both a cause and a consequence of cellular stress, interacting with other hallmarks of aging, such as genomic instability and impaired protein homeostasis. As mitochondrial quality control systems falter, damaged organelles accumulate, amplifying oxidative stress and further destabilizing the cell's internal environment. The result is a downward spiral, in which declining energy production begets more dysfunction, ultimately culminating in cell death or transformation. In light of these discoveries, some researchers are exploring whether it is possible to leverage mild mitochondrial stress to promote healthy aging. The idea is that, under certain conditions, low levels of stress may activate protective pathways within the cell, enhancing resilience and delaying the onset of age-related decline. However, the boundaries between beneficial adaptation and harmful dysfunction are precarious, and much remains to be learned about how best to support mitochondrial health in practice. What is beyond doubt is the central role of mitochondria in shaping the fate of cells, tissues, and organisms. Their decline is both a marker and a driver of aging, setting the stage for the development of chronic diseases that are so prevalent in modern life. From the brain to the heart, from muscle to metabolism, the story of cellular energy decline is, at its core, a story about the mitochondria, the silent engines whose health determines, to a remarkable degree, our own. If the fate of our cells is intertwined with the vitality of their mitochondria, then the urgency to preserve and restore these microscopic powerhouses becomes unmistakable. The story of mitochondrial decline, with its cascade of consequences for energy, health, and aging, naturally leads us to ask, what can be done to safeguard these organelles and, by extension, our cellular vigor? While the challenges posed by mitochondrial dysfunction are formidable, a growing body of scientific research is illuminating a range of strategies, some simple, some cutting edge, that may help revitalize these engines of life. Perhaps the most accessible solutions are rooted in daily choices. Nutrition, for example, stands out as a central pillar of mitochondrial health. Diets rich in plant-based nutrients and antioxidants offer a protective effect, shielding mitochondria from the relentless assault of oxidative stress. Evidence points to the Mediterranean diet as particularly beneficial, not only for heart health or longevity, but specifically for its ability to curb mitochondrial damage and inflammation. This style of eating, abundant in fruits, vegetables, whole grains, legumes, healthy fats, and moderate amounts of fish and wine, appears to support mitochondrial function at a molecular level. Researchers have observed that such dietary patterns help reduce the activation of inflammatory pathways and preserve mitochondrial integrity, providing a shield against the gradual decline associated with aging. It is not only what we eat, but also how we eat, that shapes mitochondrial resilience. Several dietary interventions have attracted scientific attention for their potential to influence mitochondrial physiology. Caloric restriction, for instance, has been shown in animal models to enhance mitochondrial efficiency, foster their renewal, and reduce the accumulation of damage. The mechanisms are complex, but seem to involve the activation of cellular pathways that promote mitochondrial biogenesis and repair. Similarly, ketogenic diets, high in fats and low in carbohydrates, may encourage mitochondria to shift their energy metabolism, potentially improving their function and resistance to stress. Increased intake of omega-3 fatty acids, found in fatty fish and certain plant oils, is another promising approach, as these fats incorporate into mitochondrial membranes and may improve their fluidity and performance. It is important to acknowledge, however, that while these dietary strategies are promising, the evidence is strongest for their role in supporting general mitochondrial function and countering age-related decline, rather than treating specific mitochondrial diseases. No specific drug has yet been approved that directly targets mitochondrial dysfunction in otherwise healthy aging, although dietary modulation remains a promising area for future development. Still, the principle is clear. The food we choose can either erode or fortify the energetic machinery within us. Lifestyle extends beyond the plate, and among non-pharmacological interventions, physical exercise stands out as a powerful regulator of mitochondrial health. When we move, whether walking briskly, cycling, or engaging in resistance training, our muscles signal for the production and repair of mitochondria. Exercise activates quality control mechanisms, prompting damaged mitochondria to be recycled and new ones to be synthesized. This not only increases the overall energy-producing capacity of cells, but also enhances their ability to withstand stress and recover from injury. The benefits are especially striking in skeletal muscle, where regular activity has been shown to delay the mitochondrial decline associated with aging, thereby preserving strength and function. Sleep, too, plays a crucial role in this intricate dance. Although the precise mechanisms are still under investigation, it is increasingly evident that restorative sleep supports mitochondrial dynamics and function. Conversely, poor sleep may disrupt these processes, amplifying the risk of dysfunction. In this way, the basics of self-care, eating well, moving often, and sleeping soundly, become profound acts of cellular stewardship. Alongside these holistic strategies, researchers are exploring a growing array of targeted interventions. Dietary supplements, for instance, have garnered attention as potential allies in the fight against mitochondrial dysfunction. Compounds such as coenzyme Q10, an essential component of the mitochondrial electron transport chain, are being investigated for their ability to enhance energy production and protect against damage. Some early studies suggest benefits for certain mitochondrial disorders, though results remain mixed and more research is needed to establish efficacy in broader populations. Other supplements, including specific antioxidants, are under similar scrutiny, with the goal of bolstering the cell's natural defenses against oxidative harm. For those facing inherited mitochondrial diseases or severe dysfunction, the therapeutic front is advancing rapidly. Several new drug therapies are in clinical trials, targeting the unique molecular mechanisms underlying mitochondrial disorders. One such agent, elamipretide, is being evaluated for its capacity to stabilize mitochondrial membranes and improve their function in patients with primary mitochondrial disease. Parallel efforts include gene therapies designed to correct or compensate for genetic mutations in mitochondrial DNA, a formidable challenge given the double membrane barrier and unique genetic code of mitochondria, but one that is beginning to yield promising results in laboratory and early clinical settings. Emerging techniques in gene editing and enzyme technology are opening new doors. Scientists have developed methods to precisely alter mutated mitochondrial DNA in patient-derived stem cells, enabling both the study of disease mechanisms and the development of personalized therapies. While these approaches remain experimental, they represent a paradigm shift in the ability to address the genetic roots of mitochondrial dysfunction. In the future, such technologies may allow for the correction of mutations once considered irreversible, potentially transforming the outlook for individuals with mitochondrial diseases. Another frontier is mitochondrial replacement therapy, in which healthy mitochondria are introduced into cells to compensate for defective ones. This strategy, though still in its infancy, holds promise for preventing the transmission of certain mitochondrial disorders and for restoring function in damaged tissues. Coupled with therapies that activate mitochondrial biogenesis, regulate the selective removal of damaged organelles, and fine-tune mitochondrial dynamics, these approaches hint at a new era of tailored medicine, one in which the specific needs of an individual's mitochondria can be addressed with unprecedented precision. Of course, science rarely moves in a straight line. While the momentum is undeniable, many questions remain. How best to combine lifestyle interventions with targeted therapies? Which patients will benefit most from emerging treatments, and at what stage of disease? What are the long-term consequences of manipulating mitochondrial genetics or dynamics? These are the puzzles researchers are now working to solve, with the hope that their efforts will translate into longer, healthier lives for all. In the meantime, the take-home message is both hopeful and empowering. Mitochondria, once seen as passive engines, are now recognized as dynamic, adaptable, and responsive to our actions. By making thoughtful choices, nourishing our bodies with a Mediterranean-style diet, engaging in regular exercise, prioritizing restorative sleep, and staying abreast of new advances in therapy, we can support the very powerhouses that sustain life. The journey to revitalizing mitochondrial health is ongoing, but the convergence of lifestyle wisdom and biomedical innovation offers a path forward, one that honors both the ancient resilience and the modern potential of these remarkable organelles. As research continues to unfold, the prospect of mitigating cellular energy decline and enhancing vitality grows ever brighter, reminding us that even at the smallest scales, the choices we make can echo across the years.